Welcome back. Now I've given you an introduction to both evolutionary taxonomy, the classical means of classification, and phonetics, which may not live on in its old form in the systematics literature, but certainly it thrives in molecular biology and ecology and similar sorts of fields. Before we go on to the next school of taxonomy, of classification, phylogenetics, I want to take a step back and talk about the characters that we use as the basis for the studies that we're talking about, for classificatory studies. First of all, I guess you might have a question about the reading. I reviewed the Stevens paper, and as I'll be explaining today in this video, he kind of gets ahead of our game a little bit, and so I think I'm just gonna pull that and not deal with that paper in terms of the discussion for today on characters and homology. So don't worry about that if you're wondering where it disappeared to on Carmen. And as for that title, you're probably thinking, what the heck does that title mean, the duck test? Well, I guess I first heard this sort of saying back in the old days of Ronald Reagan, where, of course, Ronald Reagan was very anti-tax, and there was a kind of a workaround, shall we say, a way of expressing things by which the government would raise money. They would, instead of calling it a tax, which was a nasty word in those, still is in these days, people would talk about user fees and these kinds of euphemisms for getting money from the public to go into the government coffers. And I don't know if Reagan himself actually said it or one of his minions, but they would describe these euphemisms for taxes as the duck test. That if it looks like a duck, swims like a duck, and quacks like a duck, then it probably is a duck. I actually, in researching this, found that phrase goes back a long ways before Ronald Reagan. Talk about whether you're a communist or not, or all sorts of things. But in our context for today, if it looks like a duck, swims like a duck, quacks like a duck, then it probably is a duck. Gets to the basic question of sameness. How do we recognize that two things are, in some ways, in the important ways, the same? So, when people get into classifications, you undergo a study well, today these are usually couched more in terms of, can I understand the relationships among the organisms? And then you might want to translate that into a classification. Either way, if your objective is a classification or classification is a byproduct of the research that you're doing, the workflow is pretty similar. First of all, you have to have a group of organisms that you're interested in, and perhaps this discussion here, you will recognize that is kind of mirrored in the phylogenetics project that you're going to be working this first month in labs on. So you choose a group of organisms that you're interested in. And then if your question is, how are they interrelated, or how should they be classified? Well, what are the data that underlie your attempt to come up with an answer to those questions? Our data are basically characters. And I talked a little bit about what characters were in the last video, and it's a very, shall we say, broadly defined sort of thing, that anything can serve as a character. We are interested only in characters that are heritable, that are part of the genetic makeup of the organism. And frankly, we're learning more and more these days about other aspects of heritability that don't specifically have to do with protein coding genes or RNA coding genes or things like that, but epistatic interactions, control factors, about transcriptional control, and these kinds of things that could also be used as characters. But these are heritable in a sense. They're not the result of outside influences. That's not what we're after. We're after we're looking to find characters that are part of the organisms themselves. So they can be from the most basic things like molecular sequences up through physiological processes, structural changes, behavioral changes, any of these things, insofar as they have a heritable component, have potential to be used as characters. Now, I say have potential. You want some variability in your characters. After all, if in the taxa that you're studying, all the characters are absolutely identical, then you have no variability, you have no data. It's not telling you anything about the problem you have at hand. So you want some variability. But you also need to be cautious of too much variability. If all of your characters are different in every OTU, every organism that you're looking at, then it's not going to tell you anything other than your OTUs are different. You're not going to be able to tease out the relationships among those organisms if they don't share anything in those characters. So we want some variability, but not too much variability. In the context of your phylogenetics project, this manifests itself, this appears, sorry for the overblown language, this appears in gene selection. We know that some genes evolve very quickly and some genes evolve very slowly to the point where they might be almost absolutely identical between your organisms. So you don't want things that evolve too slowly, and you don't want things that evolve too fast, that you have no point of comparison between them. Classical examples of quickly evolving genes are, well, all the mitochondrial genes. CO1, we'll be talking about in terms of DNA barcoding. CO1 evolves very quickly. You can find a significant amount of difference between very closely related species. Also, chemoreceptor genes. These are nuclear protein coding genes, which are probably the fastest evolving family of genes in all organisms, or at least all animals. And so sometimes they're evolving so quickly, not only the sequences of them evolving, but you get lots of gene duplication and gene loss. And so actually knowing to presage the rest of this video, knowing whether the genes you're looking at are orthologous or not becomes a real issue. On the other end of the scale, you have some genes that evolve very slowly. One of the ones that turns out to be very useful for looking at higher level relationships among insects, at least, is 28S RNA. Everybody has 28S RNA, which is, you need that point of comparison, and it evolves fairly slowly, such that you can start to see the patterns of evolution that took place 100, 200, 300, 500 million years ago. And you can probably think of some other genes that are evolved slowly this way. The bacterial, 12S RNA. Pardon me if I got the number wrong, but there you're talking about being able to make comparisons that go far beyond maybe a billion years ago. Some of the homeobox genes, wingless and things like that, evolve very slowly, and so they're useful for comparison. So you want to kind of match the speed of evolution, is it the velocity of evolution, in the gene with the question you have at hand. If you are focusing on a group of very closely related species, closely related in the sense that they evolved fairly recently, then in order to find molecular differences among them, you're going to need a gene that evolves quickly. Mitochondrial genes jump to mind. If, however, you're looking for to examine the interrelationships among the orders of insects, and the modern insect orders started to arise in the Upper Carboniferous, perhaps even earlier, but definitely Upper Carboniferous and Permian eras, then you want something that's evolving slowly, so that you don't have so many changes that basically it's like a videotape or an audio recording where you just record over the sequence of interest over and over again and lose the information content there. So that kind of begs the question. If we want to be comparing characters that are, air quotes, there's going to be a lot of air quotes today, air quotes, the same, what is the nature of that sameness? We want to make sure that we're, in the old aphorism, we're comparing apples to apples and not apples to oranges. If we are looking at the modifications of the anterior limbs in vertebrates, wings, fins, legs, loss in the case of snakes or something like that, we want to make sure that we're comparing forelimbs and not inadvertently comparing the hind limbs up there. We want to know that somehow they're the same. And so the term homologous or homology is used for this. And I'm sure you've encountered this in general biology, the distinction between homology and analogy. And this turns out to be a rather, perhaps not as straightforward an issue as you might have expected. And so we have the question of what exactly is homology? What is its definition? And then once we have a working concept of what homology is in theory, how do we use that and recognize, what are the recognition criteria by which we say that two things are homologous or not homologous, as the case may be? So, the first thing I want to emphasize is this distinction between the definition of the concept and the operational criteria, the way that we put it to work in order to recognize these things. The term homology, and definitely the concept, appear long before modern biology. Definitely in the pre-Darwinian period. I even saw one reference that Aristotle was talking about homology back thousands of years ago. So this is an old idea. In the pre-Darwinian, pre-evolutionary context, it was recognized, that we wanted to be able to be looking at the same organ. In those days, they were concerned with structure, right? That's really most of what they had to work with. The same organ in different organisms. What causes that or what is that sameness depends upon the basic philosophical background you're coming from. So if you're in the pre-Darwinian context, that sameness might be due to the fact that what we're seeing in the everyday world are manifestations, instantiations of some Platonic ideal, some archetype, some archetypical forelimbs in a vertebrate. But as we get into the evolutionary era, I think it's safe to say that the concept of homology becomes intimately tied in with the idea of evolution, of descent from common ancestors, and that two structures that are shared in different taxa are said to be homologous if they have inherited, if those two taxa have inherited those structures from a common ancestor. Obviously, if you're a phoneticist, this whole concept of homology and inheritance from a common ancestor, it might be fine and dandy in a sort of chat over a beer in the bar, but because evolution itself or evolution as it took place tens, hundreds, thousands of millions of years ago cannot be directly observed, then they don't even give any air, any oxygen to the concept. That's a non-starter to them. So you might ask yourself, well, how do phoneticists actually, if comparison of the same structures is the source of the data by which we do classification, by which we do systematics, how does a phoneticist do this if they don't believe in homology? Well, one way is to look at something like this, where if you're studying a group of organisms, in this case these fish, it's not unreasonable to say that I can recognize, I can confidently recognize landmarks on the overall structure of this fish, such as what is 1, the snout, 2 is the eye, 3 is the supraoccipital crest. I guess that's where the head stops and the spine begins. 4 is the origin of the dorsal fin. These are recognizable, definable, and in their terminology, operational landmarks that you can use when describing the structure, the body structure of this fish. You'll still see this concept used in morphometrics today. Or maybe you're interested in differential growth patterns and how we get to one morphology from an ancestral morphology. You define your landmarks, and then conceptually you could have a measurement between every pairwise combination of these landmarks, and the sum total of those measurements with some give and take on its accuracy could be used as a description of the shape of that fish. We look at it and we see the gestalt, the bow plan, the overall shape of this fish as a mental construct. But you can express that mathematically by having a lattice of lines that are connecting each and every one of those points, their lengths, and then that can express the shape of the fish. So in the concept of phoneticists, the things that they are comparing are definitionally, I think this is safe to say, definitionally precise, such that we're not going to confuse position 5 here, which is the posterior extreme of the dorsal fin, with position 6 here, the 6, 7, and 8 superior caudal peduncle, so the beginning of it. If you do run into problems with definitions there, then you're going to have some ambiguity, and so you need to refine your terms. So with that as just a short detour into phonetics, let's get back to people who do want to incorporate evolutionary considerations into their work. So, homology. Two structures are homologous if they are inherited from a common ancestor. That's the straight up use of the word homology. You will also encounter the word homology in other contexts, and here's one that's very important, and that is the concept of serial homology. Whereas homology, the word itself, refers to structures in two different organisms, and probably I don't think you'd be talking about two organisms of the same species, although I suppose you probably could, but typically you're talking about, two structures in the same species. Typically you would be talking about two structures in different species, in different taxa, perhaps very distantly related taxa, and that they inherited those structures from a common ancestor. Serial homology, we're referring to structures that are thought in some way to have the essence of sameness, there's those air quotes again, in different parts of the body, such that we would say that one of these anterior pairs of legs in the centipede are serially homologous with the posterior legs in the centipede. There's some sense in which these legs, these appendages, are the same. What the underlying essence is of that sameness is often almost entirely unknown. In some cases, we're getting a better idea of the developmental pathways that lead to the elaboration of legs on different segments here. And so we understand some of the genes that are involved in that and some of the transcription factors that are involved in that. But to say that we know exactly what a leg is here and what it shares with a leg up here is probably an overstatement. But we do see similarities between them. And so I think it's safe to say there's some underlying heritable genetic components here that make this leg, in a sense, the same as this leg back here, even though it's occurring on different parts of the body. Now, in the case of the legs on the centipede, that might be bleedingly obvious that it's no problem to say, well, yeah, the anterior pair of legs is, or The more anterior pairs of legs are very similar and therefore in some sense the same as the posterior pairs of legs. But it gets a little bit more complicated as these structures diverge in structures, such that are the antennae the same as the legs? Are they serially homologous with the legs? Are the poison jaws, this is why I was backing off of the anterior pair of legs, because the mandibles of a centipede and the poison jaws behind them of the centipede are also now believed, now I think it's safe to say pretty well known, to be serially homologous with the rest of the legs on the body, even though they have diverged to a greater or lesser degree, sometimes tremendously in their structure, such that that wasn't such a clear proposition in years back. So, homology versus serial homology. In both, we're talking about this sameness between the structures. In the case of homology, it's because those structures were inherited from the common ancestor. In the case of serial homology, it's the repetition of organs with some underlying genetic basis, some underlying heritable basis in different parts of the body. And this is typical of, as you know, of segmented metameric animals, that we get repetition of elements of the mesoderm, ectoderm, the derivatives of those embryonic tissues. Another use of the word homology that you'll run across is something that not too commonly, but people do refer to the term special homology. And really what they're doing there is equating homology with shared derived characters or synapomorphies. Don't worry about the jargon yet. We're going to be getting, this is jargon that's derived from phylogenetics, systematics, cladistics, and we're going to be getting to that shortly in the very near future when we talk about phylogenetics. So, our working definition of homology is that it's that structures are said to be homologous when they are found in two or more taxa and that share these features by virtue of inheritance from a common ancestor. So one might ask, how do we recognize homology? How do we test a hypothesis of, or how do we even develop a hypothesis of homology? In the first place you look, think of that, well, the centipede is probably not a good example, but think of the forelegs of an insect. The first thing you would look at is similarity. Isn't that what we're using in order to hypothesize that those structures are homologous? And we're going to come full circle on this. So let me start out with the negative approach to this. Similarity has its dangers. That is that there are homologous structures that can appear very different. What's a good example? The antennae of insects. You know that the antennae of insects have a great variety of shapes and internal structure, how many subsegments there are in these things. Are they very long? Are they very short? All sorts of variations. And so I think we can agree, without me having to go into the developmental processes involved, that the antennae of insects are homologous, so that the antennae of a scarab beetle is homologous to the antennae of a cockroach, even though there are many, many differences between them. So homologous structures can appear to be very different. And conversely, non-homologous structures can appear to be very similar. So similarity itself is not the be-all and end-all. It's not a defining feature of homology. But I think, as you'll see, I'm going to argue that it is an important feature of the development of hypotheses of homology. So take these two statements as a caveat about the use of similarity in order to recognize homology. Oh, here's a good example. This is a banana leaf. We all know what leaves are, all right? So I could ask you to enumerate all the characteristics of this banana leaf when it's probably about 4 feet long or 3 feet long. It's got this giant midrib going down. It's got veination that's carrying photosynthates from the mesophyll of the plant down to the rest of the plant. You got water and minerals being transported up into the plant. This is the main photosynthetic organ of a banana plant. And I think you can also agree that this maple leaf, if we ignore the galls caused by the mites on the maple leaf, that the maple leaf is homologous with the banana leaf, even though they look very different. Or maybe they look similar enough that it doesn't give you any hesitation in saying that these two things are homologous structures. We can see lots of differences between them, but fundamentally, a more or less two-dimensional structure, lots of chlorophyll in here. Here's where the photosynthesis is taking place in the plant. So those are homologs, no problem, right? Well, here's two examples that exemplify the caveats I had, 2 slides ago. One is that homologous structures can appear to be very different. Not the big green part of this prickly pear cactus, that's not the leaf. The leaves of the prickly pear cactus are the spines that are coming off here. Now we can see that these are extraordinarily different in shape, different in function. If they're photosynthesizing, it's only to a minimal extent. Most of the photosynthesis in this prickly pear is taking place in the stem here. They're not really a two-dimensional structure, two-dimensional flat structure like we saw with the leaves of the banana and the maple. Very different. And yet, we can demonstrate that these things are, the spines that is, are actually highly modified leaves of the cactus. Conversely, here's a tree fern. Now, that would seem to meet our definition of what a leaf is in this plant, and you would probably conclude that the fronds there on this tree fern are homologous with the leaves of the banana plant, the leaves of the maple tree, and here we have the leaves of the cactus vine. But in fact, these structures in the tree fern, the fossil record demonstrates to us that these structures evolved independently of the leaves of bananas and the rest of the tracheophytes. Is that the right classification? I've forgotten my plant classification already. Just taught it a year ago. But these evolved independently. So the fact that they're similar is due to the functional constraints or the functional demands placed upon these organs. They're primarily there to collect light for photosynthesis to take place. And the similarities that we see here in the fronds of the tree fern and the leaf of the banana plant are not due to inheritance from a common ancestor, but due to the response of the organism to similar environmental demands that are placed upon it. So, just to reiterate, the basic lesson being, just because two structures are homologous doesn't mean they're necessarily similar. They're going to look similar. You might have to look very deeply in order to uncover the underlying similarities. And conversely, just because two structures appear to be very similar doesn't mean that they necessarily are. So, the classical criteria by which, criteria, I think, as I tried to say this, criteria implies a level of definiteness that I think is misplaced here. What we're basically talking about, and this phrase is going to appear on several of these slides, is that so-called homology statements are less statements and more accurately hypotheses. When we say that two things are homologous, we are basically hypothesizing that they are homologous. And a hypothesis by its very nature can be incorrect, can be proved wrong. But I want, for the moment, to address how do we come to these hypotheses of homology. And these are typically referred to as criteria. And Riemann is kind of the go-to guy for articulating what he called criteria. There are 6 of them, by which one would hypothesize that 2 structures are homologous. And again, just to reiterate, when I use the word structure, I'm using that in the very broadest possible sense of that term. So I said 6. I'm only going to show you 3 at a time here, because Riemann basically divided his criteria into principal criteria and then some auxiliary criteria. So the principal criteria are positional criteria. The principal criteria are, first, the positional criterion, structural criterion, and then transitional criterion. What I mean by those terms are, if 2 structures are similar in where they occur on the body of the organism, I'm probably unconsciously going to default to thinking about animals, but this is also true in plants, in fungi, conceptually in any possible organism. So if 2 structures are in the same position, not only position on the body, but position in relation to other structures, then that is evidence that they are homologous. The structural criterion you can think of in terms of how this structure is put together. What are the parts that go into making it? When you think of an insect antenna, we have the 3 musculated segments of the antenna. And so the musculature that defines those is shared amongst organisms, and so it's evidence that the antennae are homologous. It could also incorporate the idea of the complexity. If you have a very complex structure and you can see that the parts are put together the same way, that this structure is built the same way, then that is evidence of homology. So position, it's position on the body and its interrelationship with other structures, and then the structural criterion, how it's put together. And then finally, the transitional criterion. Actually, there's 2 aspects of this. So when we think about the banana leaf versus the cactus spine, how do we know? How do we come to hypothesize? Got to be careful that those 2 things are homologous. Well, one way is that we can look at the development of the leaves from their primordia, and if we can see through the ontogenetic development of, in this case, the leaves, that they go through common process and then they diverge in their structure later on, become specialized in their structure later on. That's evidence that they have a common ancestral origin. We can think of that development through time in the ontogeny of a single individual. Or we can also think of it from a paleontological point of view. If we can find structural intermediates between them, if we see 2 different structures in modern organisms, when we find fossils in which those structures approximate one another, find an intermediate stage there, then that's also evidence to suggest that what we now see as those 2 different structures are actually, in a sense, homologous. They're the same in some fundamental sense. Let's see, I've got a couple examples here. So here we have, so this is an earwig with a picture of the earwig on the right-hand side, just to give it a touch of reality. And on the left-hand side, an earwig in which we've exploded the mouth parts out. And so you're all entomologists. I'm sure you know all the mouth parts of the insects. So anteriorly, we have the labrum, and behind that, we have paired mandibles. And then behind the paired mandibles, we have paired maxillae. which have nice structure, apical lobes, galea and Licinia, the maxillary palpus coming off, the subdivision here, and of course all the musculature that goes with that. So anterior to posterior labrum, then we have the mandibles, then we have the maxillae, and then the posterior most fused pair of mouth parts in the earwig and in insects in insects in general, but here exemplified by chewing mouth parts, is the labium. You can see that this arises as paired structures embryonically, and then they fuse as development proceeds. So that's the mouth parts of an earwig. Straightforward chewing mouth parts. And here we have the mouth parts of a mosquito. Superficially, very, very different structures. But are these the same? Are these, can we find homologs between the mouth parts of the earwig and the mouth parts of the mosquito? If we just look at the mouth parts individually, it's going to be very, very difficult to recognize the similarities that might lead us to a hypothesis of homology. But remember that first of Ramon's criteria was positional correspondence between the organisms. This illustration here is a cross section, so instead of having all the mouth parts, all the stylets here splayed out as in this drawing, here they are nestled in their resting state, and we have a cross section through that, with posterior down toward the bottom of the slide and anterior toward the top. And we can see those same positional interrelationships that we saw in the earwig. We have an anterior labium and a anterior labrum and posterior labium here. So the anteriormost mouth part, unpaired, is the labrum, got it right that time, and posteriorly we have the labium. And if you look over here, this is the labium. You can see there's actually some symmetry, bilateral symmetry here that reflects its ultimate origin as paired mouth parts, as a pair of mouth parts that have fused together. And then in, I think that's red, we have paired mandibles that are posterior to the labrum, anterior to the green maxillae. Here in the middle, of course, for all you mosquito biologists, we have the hypopharynx with the salivary channel going through it. So by positional criteria, we can recognize that the mandibular stylets here in a mosquito are homologous with the mandibles, the chewing mandibles of an earwig, or at least hypothesize that they're homologous. We can also go in and we can look at the musculature at the base of these appendages and see similarities in that between these two very, very different organisms, but that they share some things in common. What is this? Oh, this is the structural criterion. And here we have one of the classical examples that says whale down here of homology amongst very different looking organisms. And of course, this is scaled to be more or less the same size, even though in real life, these are very different in size. And we can see that structurally, they're made up of the same components. The humerus, I always forget which one is the ulna and the radius, and then the carpals, metacarpals here. And we can see that the forelimb of a human or a primate of any sort has many similarities with the forelimb of a whale, another mammal, or even the forelimb of a bird, humerus, ulna, radius, etc., as the different components. So they have structurally the same components. The relative sizes, the shapes may differ between them, but we can see that structurally they're very similar. And of course, I think you can also see that the concept of structural similarity versus positional similarity, there's a broad area where these things overlap, because we can see that the set of yellow, what are those, the carpals, are between the ulnar radius and the metacarpals down here. I apologize if I'm screwing up my vertebrate morphology. I will be more accurate with my insect morphology, I promise you. I was looking for examples of transitional sequences and looking for images to exemplify this, and I didn't find a lot of things that really struck me as being good examples. But this image is trying to show that embryonically, through ontogenetic development, there are similarities between very different organisms. And I want to focus here on what they're calling gill slits here, or the pharyngeal arches here. In birds and mammals, of course, these don't end up as functional gills in the free-living either larval or adult stages, but the same structures that you see in the embryos of fishes do end up being functional gills on the sides of the pharynx of the organism. So you can see the similarities between those gills in the pharynx of lampreys or sharks or things like that. You can see similar structures in the embryonic stages of very different organisms, and then hypothesize that the things that these develop into, such as jaws, are homologous, not serially homologous, but homologous with the structures that we find, very different functioning structures that we find in things like teleost fishes. So those are Ramon's principal criteria. His auxiliary criteria are, do these structures that you're asking yourself, are they homologous? Do they commonly occur in closely related species, what he called adjacent? Actually, he didn't call them adjacent. He wrote in German, but one of the main translations into English of this, adjacent species, kind of being agnostic as to relationships, or trying to be, but I don't think very successfully being, because we're thinking of adjacent as being related. So are these structures found in related species? And conversely, are they not found in species that we know, and we can argue about how we quote unquote know things, are they not common in unrelated species that we know are unrelated? So as you can see why these are called auxiliary principles instead of auxiliary criteria instead of principal criteria, because there's a lot of gray area here. Is it common in adjacent species? It doesn't mean that if it's not present in a closely related species that the hypothesis of homology is falsified. It doesn't mean that at all. The last auxiliary criterion, I'm going to get to a little bit, talk about in more detail a little bit later, is the distribution of your potential homologous structures, distribution among species. Is that distribution congruent with other structures? Does the distribution among species match? Talk about that in a little bit. None of these, I hope I've emphasized, and I know it's going to show up yet once more, none of these is definitive. These are all more or less rough and ready correlates of homology. So each of these provides evidence in support of a hypothesis of homology. And we have to keep in mind that these are hypotheses and not proof of them. Okay, take a step back. A little terminology that I'd like to introduce you. I'm sure that you've seen most, if not all, of these terms before, but let's just make sure that we cover all of these. First of all, I've been concentrating on the term homology. And in the general biology texts, all the ones that I've read, they always contrast homology with analogy. And so if we're talking about similarity, in contrasting these two terms, then homology is similarity that occurs by virtue of descent of the same structure from a common ancestor. Analogy, in contrast, is similarity that has evolved independently in two organisms. So the fronds of the fern and the leaf of the banana would be thought to be analogous structures. Also, we also have to be careful of what the computer scientists would call scope. And I think one of the common examples that you see of analogous structures in general biology textbooks is to compare the wing of a bird with the wing of a bat, and say that as organs to generate the aerodynamic forces that allow these creatures to fly, those two wings are not homologous, they are analogous. They did not evolve those features that enable them to generate the forces to fly from a common ancestor. They evolved independently. The scope part comes in because while as we think of the wings of a bird and the wings of a bat as analogous and not being the same, in fact, they both are the forelimbs of two different classes of tetrapods. And so if we look at those things in the context, in the scope of all tetrapods, then in fact they are homologous. They have evolved similar structures and their subsequent similarities by virtue of their independent origin as organs for flight. But in the larger scope, in the larger context, they are homologous. And so it's always important to keep in mind the context within which questions such as homology and analogy are being asked. Secondly, the terms synapomorphy and symplesiomorphy. One of the reasons that I cut the Stevens reading, synapomorphy and symplesiomorphy, that's going back to that concept of special homology. These are terms that were coined, or at least brought into prominence by Willy Hennig. Synapomorphy simply means shared derived characters, the syn at the beginning of that word and the sim at the beginning of the next word refer to the sharing of features. Synapomorphy, sharing of derived features. Symplesiomorphy, the sharing of ancestral features. We're going to get into that in more detail in the phylogenetics. But there are a number of workers who equate homology with synapomorphy. And for my taste, Stevens relied too much on that. And so that's kind of for me getting the cart before the horse. We'll talk about synapomorphy and plesiomorphy in the context of phylogenetic systematics rather than in the context of homology. If you're talking about molecules, common terms that you'll run across, people do use the term homology when they're talking about genes, homologous genes. But more commonly, what you see people use are the terms orthology and pairology, because both of those terms, with justification, could be used in the context of homologous genes. If you look at two different organisms and you say that, I don't know, their CO1 genes are orthologs between the two, what you're saying is that the mitochondrial CO1, I should specify, the CO1 gene is shared between these two organisms by virtue of them inheriting those genes from their common ancestors. So it sounds a lot like homology, right? But in the context of genes, we also have to take into account the idea of pairology or gene duplication. That is, in a single organism, you could have a gene duplicate, and then you would say those two copies are pairologous. Ultimately, they were a single gene in the common ancestor, hence the parallel with homology, but now it's duplicated, and now they're independent structures. This concept, pairology, may harken back in some ways. I haven't thought about this, so I'm a little bit trepidatious about asserting it too strongly, but it may remind you of the concept of serial homology, that they're the same in some sense, but different in other senses. So if two structures again in the broad sense of that term, are not homologous. What are they? If they are similar, they may be similar by virtue of convergent evolution. I think the leaves of the banana and the leaves of the fronds of the tree fern would be examples of convergence. So basically, you're ending up with structures that are similar, but they have converged on that similarity. They have evolved it independently of one another. And so it's basically environmental demands or the functional demands of the structures that, there's only so many ways to skin a cat, right? In order to get to that point of having an efficient light collecting organ in a plant, you're going to converge on the kind of structures that look to us to be leaves in both ferns and the rest of plants, even though they evolved independently. There is a parallel sort of concept. I guess I stumbled into saying parallel there because the term is parallelism. And this recognizes that sometimes we see in closely related organisms structures that are very similar, but for reasons that I'll get to in a minute, we might conclude that they are not homologous, that they evolved independently. But because they evolved from a common genetic background, relatively recent common genetic background, the skids were greased, if you were, to end up with structures that look similar. Whereas in the case of convergence, we're presuming that we arrive at similar looking structures from totally different genetic backgrounds. In the case of parallelism, there was some common genetic machinery there that predisposed them to end up with things that look similar. Obviously, this is going to be a very difficult thing to recognize because they're going to share lots of features, not only what the structures end up looking like, but actually the mechanisms by which those structures develop in the first place. And then the final term there is reversal. Which can typically reversal is thought of, let's say most commonly, it comes up in the case in which structures are lost. So we can imagine a structure evolving in an organism and then it's lost. Once it's lost, the absence of that structure can look almost exactly, if not exactly the same as the primitive absence of that feature. And how do you distinguish them? Sometimes there are hints that are left behind to tell you this, but often there aren't. So reversal looks an awful lot like absence in it. These three terms collectively, similarity that is non-homologous in its origin, are often collectively referred to as homoplasy. So non-homologous similarity is homoplasy, which can arise from convergence, parallelism, or reversals. Oops, go at it this way. So I promised a little bit about molecular data because that is one of the major sources of data these days. I can tell you that when I began my work in systematics, we were relying upon morphological characteristics, and you might be able to find, dozens. Perhaps if you were really good, you could find hundreds of morphological characters. But when you're dealing with thousands of species, even 100 morphological characters is not going to be sufficient, most likely, for you to figure out how all of the species are interrelated, because some of the species are going to be exactly the same, even if you have hundreds of characters. But with molecular data, we have thousands, perhaps even millions of potential characters to use. So that's been a tremendous boon for systematic studies and has just absolutely revolutionized the field. So if we think about molecular data in its largest sense, think about two genes in two organisms. Say the mitochondrial CO1 gene. I keep coming back to that example. Why do we think that those sequences are orthologous and homologous? You might argue that a hypothesis of orthology is based upon the fact that the genes have the same function. That might be true in a limited number of cases, but of course, in the vast majority of cases of genes and organisms, we don't actually test to find out what their function is. We impute, infer, hypothesize their function because of their similarity to the genes in other organisms where that function has been directly addressed. We rarely do so directly. So while you possibly could do that, in practice, we usually don't. And of course, we also know that there are multiple genes that serve the same function. There are multiple RNA polymerases in an organism. So which ones are orthologous and which ones are not orthologous is not based upon at least the function as expressed in the term RNA polymerase. Typically, what we're talking about when we say two genes are orthologous has to do with their structure. The same way that the forelimbs of tetrapods are homologous, we say that genes are orthologous on the basis of their structure. That may be because of similarity in the nucleotide sequences, or if we're talking about a protein, in the amino acid sequences. That could be one component. Another one could be the number and position of introns in a nuclear gene in a eukaryote. That can provide some hints. It could be where that gene occurs in the genome. Going back to CO1, in the vast majority of insects, CO1 occurs in the same relative position in the circular mitochondrial genome in relation to the gene that precedes it on the coding strand and the one that follows it on the coding strand. So its position within the mitochondrial genomes tells me that's a CO1 gene. It could be, and now we're getting a little bit more tenuous, that if you have primers to amplify a section of a gene, you throw the primers into a DNA extraction of an organism, and you get a single amplicon coming out, that you're going to say that the amplicons of this organism and the amplicon of that organism are orthologous because the primers match the single section of DNA and amplified it. Well, that could be evidence of orthology. It could be the result of the fact that I guess it would be bad luck, your primers matched different places in the genome and amplified different segments. So that's not a sure thing, but it's a good primer design will seek to minimize that. So probably usually you wouldn't come up with primers that are amplifying the wrong section of a genome, only coming up with a single band. You probably get double bands or something like that. So these are the kinds of things we can talk about in terms of genes being the same. To find homologous, orthologous genes within an organism. Then if we look directly at those genes, we can talk about the individual monomers within the gene. Well, within a gene, we'd be talking about nucleic acids, right? Or we could talk about the same thing in a protein in terms of the amino acids. So we could talk about individual residues within a larger sequence, them being homologous in some way. And this leads me to the subject of sequence alignments. So here I've pulled a little piece of some sequence alignments that came out of my lab. You can probably tell by the name that these are all CO1. So this is mitochondrial CO1. And this is what's called a multiple species alignment. So these are different OTUs. I actually don't know off the top of my head if they represent different species or not. But that's the name of them. And here we just see a chunk that it took out at random of the sequence. After we've aligned them, we can see there are parts of these sequences where all of the organisms, all of the OTUs in the analysis share exactly the same nucleotide here. Here's another section. Here's a shorter section. Here's a small section where everybody's got a G in the sequence. Larger, larger. Uh, so in a sense, we are saying that this residue here, this A, is homologous with this A here. And we can say that because the overall sequence alignment puts them in the same position within this within these sequences. And so in a sense they're the same thing, and in another sense they're the same thing in that they're the same molecule, or at least the same monomer here. Let's see, do I want to say anything else here? Let's move on here. This is probably pretty familiar to you. We have some organisms that are varying in there, but we would still say that this position, even though the nucleotides vary amongst individuals, this position is homologous. Not because of similarity at that position, because obviously we have some differences. We got 5 C's and 6 T's there, but because it's flanked by TG on one side and TG on the other side, and the overall context here gives us good confidence that what we're talking about here is a homologous position, that in some organisms either it changed to a T or it changed to a C. We don't know which. We don't know the direction. We don't know what they came from either, to be honest with you. But that this is a homologous position. Now I showed you that finished product, that multiple sequence alignment or multi-species alignment is basically a homology statement that, while in the context of those CO1 sequences, might have been, it's possible that you could have just given me the sequences and I could, by eye, just have aligned them. But sequence alignment, especially when you start to talk about multiple species or species that are not closely related, you start to get enough differences that it becomes fairly difficult to be honest, it becomes impossible to align them by eye unless you're some sort of savant. And one thing I used to do, that I have done in class, is to go to this website, philo.cs.mcgill.canada there, and try your own hand at aligning sequences. It's a little game that you can play online. And if you can stand the background, the very, very annoying background music that they play with this little game, you start to get a feeling for how difficult it can be to align sequences, because not all sequences are the same length. Sometimes, even if you have the same starting and same ending point, you can have sequences that differ in length. In other words, perhaps at one or more positions, there's an extra nucleotide, or there's one fewer nucleotide. These are called gaps in the alignment, because you see that there's no matching pair between them. In the example I showed you, there were no gaps. Those sections were perfectly aligned. So you can appreciate, I think, if you play with this a little bit, how difficult this process can be. Because it's so difficult, we commonly use algorithms. The algorithms that were used 20 years ago are no longer widely used. They've been supplanted by newer algorithms, and these will be supplanted by newer algorithms as we go forward. Some of the common ones you see people use today, I have no recollection of what these acronyms stand for. Everybody just calls it, I used math to align my sequences, or I use muscle to align my sequences. But this is letting the computer do the task of hopefully finding the correct alignment. Of course, we don't know what is correct a priori, so we are assessing our approach to correctness on the basis of some optimality criterion. That is, we're trying to say this is better than this by this measurement. And so whatever you're measuring is the optimality criterion. That's going to pop up again when we talk about phylogenetic analysis, optimality criteria. So let's see, what did I want to say here? So basically you're trying to match up areas that are the same in the sequences, inserting gaps into the sequence where necessary, and typically gaps are necessary to be interpreted. What we're essentially doing is looking for homologies between the positions of the sequence that are basically very simple characters, in contrast to characters like, let's say, the structure of an orchid flower, which is very complex. All these pieces and parts, and they're put together in a particular way. Then you can look at one flower and look at another, and you can say, these are homologous structures because of all these pieces and the relative positions and such. When you're talking about DNA sequences, you're talking about almost the ultimate in simplicity. In a DNA sequence, aside from occasional oddballs, basically we're talking about there's only 4 things that at a position in a sequence, only 4 monomers that can be there, either A, C, T, or G. And one A looks exactly like another A, is exactly the same as another A. So it's impossible to tell on the basis of what that molecule looks like whether it evolved to be in that position independently or not. It is the same thing. Protein sequences are a little bit more complex because they have 20 amino acids to use as the building blocks instead of 4. And those amino acids have some structural, some chemical features that help, their relative hydrophobicity, these kinds of things. To some extent, this is also true in nucleic acid sequences, but to a smaller degree. We get into it with RNA sequences that loop back on one another, and whether they are capable of pairing in the stem regions. But I digress. Alignment itself, alignment itself can be trivial, meaningless. If when we go along inserting gaps, there's no cost to doing so in terms of our optimality criterion. If we're going to say one criterion for optimality is the perfect matching of nucleotides along the entire sequence. Well, we can make that perfect match if we just insert gaps wherever we need them. Say, oh, this one needs an extra little gap here, so I'll push this over until it matches. And so that if we don't have a price for putting a gap into our sequence alignment and for making the gap bigger or smaller, there's no price to be paid, then alignment is trivial and meaningless. And so we need this balance between the number of let's talk about DNA, the number of DNA bases that match versus the number and the size of the gaps that we have to infer in order to make them gaps. So basically, this becomes something upon which we have to rely upon computer algorithms to do. And just because we run it through an algorithm doesn't mean that we get the correct, the best. Doesn't even mean that we get the best alignment. It's just that we get the alignment that we were able to be satisfied that was calculated in the amount of time we had available. In a larger sense, though, we're trading off simplicity in characters, A versus T, where we could have convergent evolution, reversals that would be totally unrecognizable because one A just looks like the next A. We're trading that off for quantity, that we have thousands or millions of DNA markers, not markers, DNA bases in our sequence with which to compare organisms. Some of them are going to be wrong. Some of them are going to be convergent evolution. Some of them are parallel reversals. But we are banking, knock on wood, that in the aggregate we're going to get more informative than uninformative characters in this process. So let's see here. So ultimately, a hypothesis of homology is based upon similarity. Even though I gave you the caveats at the beginning of the discussion about how similarity can lead you down the wrong path, ultimately the underlying basis for hypothesizing homology is similarity, and features like the complexity of the structure, the relative positions of its pieces, what it does, and so on and so forth. These can all be reassuring, but they are not dispositive. They don't prove the case. Ultimately, the basis for the hypothesis is similarity. But now I'm getting to Ramon's last auxiliary criterion, the one of congruence. If we are focusing our attention, if we're interested only in vertical transmission of characters, that is, transmission from one generation to the next through time, or from one species to the next as that species, that parent species speciates into two daughter species, if we limit ourselves to that vertical evolution, then first of all, we expect the distribution of characters to be hierarchical, that the daughter species inherit their homologous characters from their parent, and only from their parent. Whatever that parent evolved doesn't show up in some other lineage. That would be horizontal transmission, but is limited to its descendants. So we expect to find that the characters are distributed hierarchically. And we would therefore also expect that different characters that are limited by the same history of speciation, this ancestor-descendant relationships, they're both coming from that same source, that different characters will also conform to that same hierarchy. That is, they will have congruent distribution among organisms. And so this is kind of a test, if you will, of your hypotheses of homology. If you take your sequence, let's say you've got some CO1 sequences you're going to run a phylogenetics program on, and you get a branching pattern. Then if you go back and look at the individual characters, the individual states there, characteristics that don't match the hierarchy that you come up with, that you hypothesize, are in a sense, well, these misfits are falsified hypotheses of homoplasy. It may have met all the previous criteria of Ramon or whatever, your sequence alignment, whatever, but the fact that they are not congruent with the other characters suggests that your initial hypothesis of homology was incorrect, that you may have parallel evolution. So this gives rise to the idea, this is another Hegelian term that we'll get to, the idea of reciprocal illumination. That if you take your data, your initial hypotheses of homology, infer a pattern of phylogenetic branching on the tree, go back and look at your characters, find the ones that don't match that branching pattern, and then perhaps reinterpret those. Perhaps what you saw as a leaf in a fern tree, the rest of the data suggests that no, that's not a leaf, because the distribution of leaves amongst all these taxa just don't match the phylogeny. It looks like those leaves evolved separately. Reciprocal illumination, meaning that you start out with a hypothesis, you come to a conclusion, and now you go back and look at your hypothesis in the light of that phylogenetic pattern you discovered, and change your initial homology statements. I think ultimately if you do this to its extreme and you just recode your data, then you're going to end up with perfect phylogeny, and obviously that's not really what we're after. We're looking to understand the sources of the non-homologous the incorrect hypotheses of homology that were made in the first place. Is this circular reasoning? Some people argue that it is. I would argue that no, it isn't. I suppose if you did this and you presented your conclusion after these steps involving reciprocal illumination as your initial position, then that would be intellectually dishonest and represent circular reasoning. But it doesn't really, because you could pull your incorrect characters from the analysis and then run the analysis without them. And that analysis will then tell you how to interpret the data from the characters that are not in the analysis. So there's an argument that it is not circular reasoning that I find convincing. Let me close with just a couple of notes on the phylogenetics project. I was there virtually in the lab, but sometimes it was difficult for me to hear, and I interpret that sometimes it was difficult for you to hear me. So some things came up that I just want to clarify my position on. The first was the question of complete genes versus incomplete genes. I think that what you're going to find, unless you limit your analyses to organisms that have complete genomes or complete transcriptomes or something like that, that the vast majority of data that you're going to find are partial genes, incomplete genes. This is not a bad thing. This is a reflection of the fact that people are using primers in order to amplify chunks of DNA. And the primer rarely, probably almost never, corresponds to the beginning and the end of the gene. The gene might be too long. Most genes are starting out with things like ATG, so already you have some trivial matching there for your primers to bind to, and they're also going to end with stop codons, and so you're going to have some trivial matching there. So the fact that they're partial genes is not really that important. What's important is that you have a long enough sequence. 2 bases is not going to be enough to really conduct a phylogenetic analysis. We're looking for hundreds of bases. Secondly, despite what the instructional videos on the Mega website say, at least the one I listened to, BLAST does not find homologs for you. What BLAST does is find similar sequences. Here we are again, similarity, homology. They may well be homologs or they may well be homologs or orthologs, but they may not be. You may have similarity in sequences due to functional constraints on those sequences. For example, if you have transmembrane proteins, then there are certain physical chemical characteristics you want in the amino acids in those transmembrane proteins, which may be reflected in similarity in the DNA sequence that codes for those proteins. You may have paralogous genes that are going to be similar, but not orthologous. So BLAST is basically, in a sense, a phonetic method. It's just finding similar proteins for you to consider as orthologs. I think someone asked about using different strains of the same organism as OTUs. Yeah, you could do this, but recognize that the phylogenetic methods that we're going to be using are designed to be looking at relationships among organisms above the species level, or at least at the species level, between which there is no gene exchange. So it's assuming vertical transmission of features. If you've got horizontal transmission going on, you're going to get some results that are going to be ambiguous. This is a real factor, for example, when you talk about closely related species of plants where there is hybridization or introgression going on, and you have what this is called reticulate evolution, where you have 2 branches that don't remain separate, but they actually come back together, and you have gene exchange. So you can do it, but expect to find some ambiguity due to the exchange of genes between strains. And then finally, just to be clear, I wanted to try this semester having you work in small groups so that you could get some feedback. So you could talk to one another, feed off of one another's insights and perhaps confusion on some sense, hopefully one leading the other out of confusion and not both leading to greater confusion. But at the end of the day, I want the reports to be written separately. So just to quickly summarize, the comparison of features is critical, is fundamental to the science of systematics. Sometimes systematics is simply referred to as comparative biology. But we have to have a basis for comparing two things. And so homology becomes important for us. We want to be comparing structures that are inherited from their common ancestor. That's our point of comparison. I suppose if you're into physiology, straight up physiology, and looking at function, you might be interested in different solutions to a common physiological problem. And in that case, you'd be looking for things that have the same function, but could come from different origins. But in terms of systematics, we're almost entirely interested, leave myself a little escape hatch there, almost entirely interested in things that are being compared because they are inherited from a common ancestor. Defining homology and identifying it, just like the concept of species, are two different things, and so we need to keep those separate. Ramon's criteria are kind of the fundamental base upon which most of these hypotheses, at least once we get away from molecules, but Ramon's criteria are a fundamental basis, with the principal criteria being structural similarity, positional similarity, and then be able to identify transitions between two apparently quite different states. Once we get into molecules, then we're relying upon the algorithms associated with multiple sequence alignment, and those will continue to develop, but they're the tool that we are using to hypothesize homology at that level. And I guess finally, just to reemphasize yet again that homology is a hypothesis. Under all the covers, at the very base, it's based upon similarity, a hypothesis of similarity, and that can then be tested, supported by congruence. That is, we expect, if we have correct hypotheses of homology, then different characters will be congruent with one another, that they can both be mapped to the same hierarchical structure, because that's how traits are passed down through evolutionary history. So next time we'll jump into phylogenetics and the world of Willi Hennig. So until then, take care and stay healthy.